on one project, but try to give an overview of several projects that have been going on in our group and in our collaborations. So in the spirit of trying to get into touch with more researchers and helping establish these networks Silvina was talking about, just giving an idea of what's going on in the groups by quoting some examples of recent research we have done. Naturally, I don't have the time to go into depth into this, but so I just want to give an overview. Whether that has been a good idea or not, you will see afterwards. So let's try. So this is where we work. The institute is a relatively small institute founded 40-something years ago. It's housed in this old house. It's funded mainly by CONICET and by the University of La Plata. It's about 25 researchers and 45 or something staff. It was established by the joining of two groups, one doing physics of liquids, and groups doing biophysics. And now there are groups doing condensed matter physics, strongly correlated systems, liquids and soft matter. There's a science education group, and then there's our statistical biophysics group. The group composition changed a bit in the last few years, so presently we are 3 researchers working permanently in the institute, and this time there are 2 PhD students. Carla is around here somewhere, and Christian. There's also an undergrad that's perhaps finishing today. So there's not many of us in the group, so the results I will go over with you here have been done through several collaborations with different institutions. Mostly the oldest perhaps collaboration that's been going on is the first one with the Cavagna and Giardina at the Sapienza University and the Institute of Complex Systems in Italy. Then we started the collaboration with Pedro Lowenstein at the University of Michigan at Ann Arbor. And we also have collaborated with people in the University in Montevideo, Hugo Fort, Osvaldo Chara, which is at Nottingham now, Dante Chialvo in San Martin, and several people, in particular Sergio Canas in the University of Córdoba. So let me just show you a few examples of the things we did and things we are doing, so maybe you can see the types of problems we are considering and the type of techniques we're using. One of the long-standing lines, perhaps the main line in our group, is flocking and swarming and collective animal behavior. So looking at groups of animals that move and then move together. We have studied through this collaboration with the Italian group, groups of birds moving together and groups of insects, midges, flying together. And in this collaboration, so one thing that emerged that I was talking about in the school is that swarms, so the difference between swarms and flocks is that flocks are ordered, individuals that fly together and go somewhere, whereas swarms are just insects that are moving or flying but are just hovering around a spot in some reference spot, okay? So they don't go anywhere. So, but it turns out that this disorder movement actually can be interpreted as a critical state. So as the group is not ordered, but on the brink of ordering, and you can see that through space velocity correlations. That led us then to study the dynamics of the groups, and we found that the dynamics, the scaling hypothesis for the dynamic correlation also holds in those groups of animals, but when you do the measurements, the critical exponents do not belong to any of the known universality classes in dynamics. So what we did, so this is a long path that started with the first article here and ended in the second one. So we tried to study the ingredient, the missing ingredient that would lead to have the correct critical behavior. And so in the end, what we did was to study field theory, which is it looks more complicated than it is. I mean, conceptually it's not that complicated. So this here is a model. It's basically a Landau-Ginzburg model, which has an additional variable, non-critical variable, fast, which is the spin. And the thing with this model is that the spin, so this variable, the spin, is generating the rotations of the other variable, the other field, which is the velocity field. These kind of models were studied in the 70s. I mean, these models were a field that's generating the rotations of the other field. But so what we did is to take these models to make them active. So we added when we study the equation. So this these dynamical equations are more or less standard. You get them from the Poisson, from these Poisson brackets, and using this Hamiltonian, you get this part of the equation. But in the left side, we replace the time derivative by the material derivative, which is telling you that this actually, the field, this velocity field, is actually making the particles move. There's actually some movement here. So the complete problem needs also a density, a continuity equation, but it makes the problem too complicated, so one can impose this incompressible condition, so to get rid of one of the fields, and you can work with this with these two remaining fields. The theory still is technically quite heavy, complicated to manage, but in principle you can use the randomization group techniques with doing perturbations, and you can compute the randomization group flow, and you can compute compute things like this, and you can compute the new critical exponents. And so in the end, we could show that this model, so this is the experiment, this is our simulations of the model, and these are the theoretical results. We could show that this model indeed gives you the correct critical exponent, which is lower than the critical exponents previously known. And an interesting thing here is that you can and you should study the crossovers because it's very important. If you just focus on the long scale properties, as one usually does in condensed matter physics, you don't get the right exponents. You get so the strictly universal exponent is different from the one we measure. But the point is that the system we measure are finite size. And so if you take into account the fact that the systems are finite, then you find that there's a crossover in the randomization group sense between two values of the exponent. But when you are studying, so when you study finite systems, so you see this is the flow. So the group, the randomization group flow stays for a long time here, and only for very, very large systems will continue and flow to the stable fixed point, which is basically the fixed point that describes the Wigzig model. Okay. So that's one of the lines. More recently, we started collaborating, analyzing experimental data from cultures and ex vivo explants from cancer, from brain cancer cells, from glioma. And this is a project that's starting. It's going on. Up to now, what we could measure, we started measuring correlations, and we found the intriguing result for us, which we still cannot explain why it happens, but the fact is that velocity correlations in these cultures and these cells are long range. So you can, if you study correlations of patches of different sizes within the system. You find that the correlation depends on the size of the patch, as this figure shows here, and this is without global order. Cells are going back and forth; they are not moving together, but still the correlations are long-ranged. So they look like also like near a critical point, but we don't exactly know how or why this happened. What would be the advantage for the system to be in that case, or whether there's some subtle out of equilibrium effect that could explain this behavior. So, but this is something which is still going on. We also did something with the ecological data. We found this, so in collaboration with Hugo Ford from Uruguay, we found this interesting data, is people getting, doing census of positions, size, and species of trees in the Barro Colorado Island in Panama. So it's a large tropical forest. And these people have been making census every 5 years for since the 80s. And it's interesting, you get, this is a 50 hectare plot, and you have lots of information about the species that are living and are growing there. That's about 300 species you can find. So it is a very diverse ecosystem. And we started studying basically correlations, and among other things, one of our results was trying to say something about the dynamics, which is difficult because it's something that moves very slowly, and we have only a few snapshots, like 5 or 8 snapshots. But it turns out that the system is quite dynamic. If you look at the abundances of the species, they fluctuate a lot. There are some species that mostly almost disappear in the plot. Some others are stable. Others grow a lot, and so on. But if you define correlation function that can compare the snapshots comparing the species content, so the species abundance, you can define an overlap or correlation function. And what we see is that the dynamics is stationary. So so this is how the correlations decay in time, but if you plot them as a different time difference, they basically give you one universal curve. So it seems that the system is changing, populations are changing, species, the abundances are changing all the time, but they're doing so in a way that's kind of self-similar in time. So we haven't seen any catastrophic trend in this system at this resolution, at least. So we have also done some work. This is with the guys in the UK. There was some theoretical work exploring growth models. These are mean field growth models. The interesting thing here is that this is a non-Markovian model where instead of assuming that a cell or an organism can just reproduce at a constant rate, you introduce a period where the cell cannot reproduce because it's growing. So when the cell is very young, it just cannot divide again. So if you introduce that, that introduces a non-Markovian growth model with some, which has several mathematical complications. But we managed to solve this model, at least in some cases, and we could, for example, get this behavior where you can see this, that the growth proceeds in steps. These steps are the time, so they reproduce, but then they cannot reproduce for a while, so, and then they can reproduce, so you get growing in steps. And we could compute that, and we could also compute the effective rate of growth. So for very long times, the thing gives you just an exponential growth, and we could compute the effective rate for that growth. Then in our other works, we are also interested in active matter, particularly with relevance to biological systems. So this was a local collaboration just with the local folks over there, numerical work, and we show that you can apply the short-time dynamics techniques to study active systems. So short-time dynamics, the idea is that when you are near a phase transition, a critical point, you can say something about how the order parameter grows when you are out of equilibrium. So you start from a completely disordered system, and you let the system evolve. The order parameter and the variance will evolve in a certain way with certain power laws if you are just at the critical point. And we found that this also happens if you are at a spinodal point. So you can also describe spinodals without needing to equilibrate the system, just by the short-time dynamics. You can say whether you are at a critical point or maybe at a spinodal. So we had done this with equilibrium systems. We tried this. We thought it would be nice to try what happens with active systems. And in fact, we could do this. We did it for the Vic model with the vector noise, which is known to have a first-order transition. So we could describe the transition both with the usual Binder cumulants and through short-time dynamics, and we could identify here. So the presence of these two spinodals, and this is the transition as identified from the Binder cumulants, and you can show that here. This is actually very compatible with the first order transition, where you can see the spinodal, and you can see that there's the actual binodal here. So the transition measured by the Binder cumulants coincides with one of those of the limits imposed found by the short-time dynamics technique. So finally, just very briefly, we also collaborated with the San Martin group on the dynamics of neural systems, or using published data from a North American group. Again, we looked at correlations, time and space correlations, and found this is a neural activity in the visual cortex of mice that were exposed basically to a blank screen. And so we studied this activity. We found that, again, depending on the size of the window you look at, you can find a correlation length that will span the whole window of your observation, the growth with the window, and also the correlation time is growing with the size of the window, again pointing to possible critical-like behavior, critical-like dynamics for this system. So to finish, then, just want to list our current interests, which are collective behavior of big animals, including humans, including pedestrians. So there have been some collaborations in the group for studying pedestrian movement, cell movements in tissues. We are starting to collaborate with people that have experiments on healing, on the damage, tissue damage, and healing. Active matter models, biologically related. We are interested in studying phase transitions and critical phenomena in biological systems, in analyzing experimental data with these techniques. And finally, some work on ecology, and well, I mentioned the forest, and there's also something I couldn't mention, but one of our colleagues is studying some effects of pesticides in movement in amphibians, in collaboration with an experimental group also in La Plata. So to finish, I will just show you these 4 images. there's a story behind each of the images. I won't tell the stories, I won't get into this. I only want to point out that there's a lot going on in my country, there's a lot more going on in the world, and the stories related to these things affect the way we live, affect the way we work, and I just cannot pretend that they are not there. So, thank you very much.
Thank you. Thank you, Tomás. And so there is room for questions.
Just a question. How many faculty in physics are doing things related to biology in La Plata?
Well, the departments of physics, it's about, I don't know, 50 professors or something like that. The numbers I gave is just for our institute, which is a small part. So the larger part of the physics researchers are in another institute, which is much bigger than that. No, not in biology, I'm sorry. This is 50 people in physics. No, in biology, I don't know. It's a handful, I don't know. Maybe professors, like, in the 10 dozen, you say a dozen, something like that,
Arthur.
Thanks, Tomás. Can you speculate about the evolutionary benefits of those brain cells having operating the critical point?
Yeah, I'm not sure. It could be, well, these are tumors, so these are not healthy cells. These are cancer cells, and these are growing, and these want to move. So then what is known is that the way these tumors grow is just infiltrating the tissue. These are not tumors that tend to travel very fast, so typically they don't do metastasis, but they are very, very aggressive. They grow a lot, and even when they are chirurgically, removed, they tend to regrow. It's like any stray cell that stays there tends to come back. And so the speculation is that this has to do with the ability to infiltrate neighboring tissue. But exactly how, I don't know.
Hi.
Thank you, Tomás. For the bees, it's very important, the swarm. So you have some...
for the bees?
Yes, for the abelhas.
Yeah. No, that's... I don't have anything on bees. There's some colleague that started doing that. But I don't know. I expect this could be very different because they are different, because they are pretty much organized in very specialized ways. And probably this kind of models we have been studying will need to be modified because they are doing very specific things, But I haven't looked at all at them.
For people that is...
Yes, absolutely. Yes.
You can help it, but...
Yes,
okay,
absolutely. It's an excellent point. Yeah.
Okay. Thank you.
Yeah.
Here. Hi, Thomas. Here and here.
Yeah.
So about your work with forests in Bajo Colorado, Panama,
Yes.
right? So I think it's the same data set that Hubble work
Yes,
in
that one.
In his, okay. But what I remember from his theory, so he used just in a given time, he didn't consider over time how those abundance would change. And did you compare so what he did is to compare the histogram of abundance when you rank it from the most abundant to low abundance. And did you see if this curve changed over time, if the slope of this? Did you compare your study with Hubble study?
Yeah, well, we had the study that I now remember. It's a bit older, and they used the theory, the neutral theory, to estimate a relaxation time. But their time was huge, was much, much larger than our estimate. At the time they published the paper, there were fewer data points, and they used this theory which makes some assumptions. This is not theory, which is only took these 8 snapshots and computed the time correlation with those snapshots, and we see that it is the gain, and we got a time at around 15 years for the decay. So much faster. They say like something like 1,000 years or so.
So your Markovian, so you also have a Markovian model behind? Did you show results with Markovian? So there's no model over there.
Not for the Barro Colorado is not a model. It's just the data
Just
and the
data.
correlations.
Okay,
Yeah.
okay, thank you.
Okay, Tomás, here. Thank you very much.
Thank you.
My question is related to Paulo's, but in the neural aspect. Usually when an animal is behaving, you have like a low-dimensional collapse, I guess, to generate low-dimensional motor patterns. So the question is, when do critical states arise, and what will be the purpose of having them?
Well, the hypothesis is that the critical states arise at rest, when the brain is not processing anything. It's just like ready to do something. And yeah, my guess is that when you, in fact, these data, for example, the mice are just looking at the blank screen. So they're not processing things, and partly. So the idea is that the critical state makes the brain responsive, maximally responsive, it generates a lot of complex pattern, and makes you able to respond in its maximum. So it's like very susceptible, if you wish. So you can respond in various ways. What I expect, and I think there are some data about that when you do something. So when you say start processing something, then you go out of criticality. So you can go to the overexcited state, to the less excited state. But so.
I like the argument, but it's very expensive to be in a critical state because you have to be generating all the time.
Well, I agree. So you're asking, why don't you just turn off all the neurons and wait? Well, yeah, I don't know, but I think that Kelvo had an estimate of what's the energy the brain must spend to be critical at rest. I forget the numbers, but yes, you must spend. So there must be some purpose on it. Otherwise, you wouldn't do that. I don't have a definite answer to that. But yeah.
Is there any other question? Okay, so if there are no more questions, did you want to? Yes.
It's actually a very quick comment. So you like to study physics of flocking, right?
Yeah.
And there is this group in Porto Alegre, the group of Leonardo Brunet, doing a lot of physics of flocking now for cells. So I think it would be nice to have some sort of proximity between
okay, yes,
just
sure. Yeah.
To mention it, yeah.
Okay, thank you. Thank you for that.
Just another comment about the data of Barro Colorado Island. I would like to ask you, you measure the correlation at the level of a single species dynamic population?
No.
Or at the level of...
No, this is the whole species. So we compare, so for the whole plot, or perhaps for a small plot, we compare how many individuals for some species are there at two-time snapshots, and we compare all of the species that are present there. Okay, so not just a single species, because with a single species it's not stationary, because some species just die, others.
Yeah.
If you get the whole ensemble of species.
But the correlation, how do you measure this? This correlation at the level of the community?
Oh, I can write it down for you of the coffee break, right?
At the end, is a measure of the community correlation in time, not of the single
No, it's a community.
species.
It's a community.
Okay,
yes.
thank you.
Okay. So thank you. I think we have to
Okay.
move on. We thank
you.
Tomás.